Hello everyone. My name is Itamar Ronen, and I am based in Brighton in the UK.
Your
And I
volume
want to
is
thank
a
the
bit low, sir.
organizers for
Can I increase the
inviting me to
volume?
give this talk on magnetic resonance spectroscopy, and
Yeah,
to you
that's
for
perfect.
listening to this talk. I hope it will make sense to you and it will enrich your knowledge in magnetic resonance. So without further ado, let's see what we're going to talk about today. So the first thing I want to do with you is to go very quickly through some concepts of magnetic resonance spectroscopy. We will establish some terminology. We will talk about the chemical shift, which is essentially a term for the different resonance frequency for different type of protons. We will also learn how to localize our signal in magnetic resonance spectroscopy, so how to make sure that it comes from a region that we are interested in. And we will also discuss what are the type of molecules and what are the specific molecules that we can reliably detect with magnetic resonance spectroscopy. I will focus my talk on the brain and also on proton spectroscopy, leaving aside the fact that we can acquire spectra outside of the brain, in the heart, in the liver, in the kidney, in muscle, everywhere pretty much in the human body, and also look at other nuclei other than protons, which will leave for a different opportunity. Then we will talk about some applications of magnetic resonance spectroscopy in the clinics and also in clinical research. And again, the focus will be on neuro only. And then we will talk about what can maybe be defined as more advanced methods related to spectroscopy. In spectroscopy, we're looking at concentration of molecules, but we will see that we can look at other properties of molecules that can be used as markers for other properties of tissue, for example, as microstructural properties of tissue, but in a different way that, for example, diffusion-weighted imaging that you may have covered in this workshop is doing. We look at the specificity of the way that we are doing this measurement to, for example, measure inside cells rather than from all compartments of tissue. And then as the last topic, I would like to talk to you about the possibility of doing spectroscopy at low field. This is a rather challenging measurement because we are not that sensitive at low field, but still we can use some trick of the trade to get specific spectroscopic information, and we will briefly discuss how we can do that. So first of all, a reminder to us all that the signal that we have, the so-called resonance that we get from the protons that we're looking at in imaging or in spectroscopy, is a nuclear phenomenon that is a frequency that is excited and then detected with the radio frequency equipment that we have at a specific magnetic field in which we perform the measurement. And the resonance frequency that we measure, that we typically denote by the Greek letter omega, omega naught, is proportional to the external magnetic field that we exert, and also to a constant that is called the gyromagnetic ratio that is different from each nuclei to the other. And we know its value in protons, so we can calculate what is the resonance frequency that we get at a given magnetic field. So for example, at 1.5 Tesla, the resonance frequency of protons is about 65 megahertz. But then when we do this measurement, for example, in tissue, and we have two types of protons in tissue that are quite abundant. One is the water that we have, the protons that are embedded in water molecules that we have in tissue, but we have also protons, for example, in lipids. And we do this experiment in which we are looking at what is the resonance frequency of these protons, and we see that we have, for example, in this case, two distinct resonance frequencies. And the resonance frequency, we said that it only depends on the magnetic field and the gyromagnetic ratio. So how comes we have two frequencies in this case? Well, we all have to consider what is the actual magnetic field that the protons feel within each of the protons, either in water or in lipids. So for example, if we look at the protons in lipids, the protons in lipids feel the external magnetic field acted upon them, which is B naught, but there is also some shielding to that magnetic field that is provided by the electrons that revolve around the proton, that when a magnetic field is applied to them, generate a weak magnetic field in the opposite direction of the external magnetic field. So the local field that the protons in lipids feel is slightly lower than the external magnetic field that we apply, and this means that the resonance frequency that we obtain is slightly lower than, for example, what we get from water, because what happens with water is that the oxygen that is bound to those protons attracts those electrons away from the protons, leaving the protons a little bit more exposed to the strong external magnetic field applied to them, and the local field that they feel is slightly higher than that that the protons in lipids feel. So we have a small difference in the so-called local magnetic field that is caused by difference in the chemical environment, and those differences create tiny little differences in the resonance frequency that those protons, in this case in lipids and in water, feel, giving rise to two distinct resonances or distinct resonance frequencies. And now to some terminology. The difference in frequency between two resonances that are distinct between, for example, two types of protons, that of lipids and that of water, for example, is called the chemical shift. And the units of the chemical shift in this case would be units of frequency, meaning in hertz. And you have to realize that those chemical shift differences are typically in the order of hundreds of hertz if you're measuring on a clinical scanner, and much less than that if we're measuring at a low field scanner. But just to give you an idea that the resonance frequency itself is in the hundreds of megahertz. So we have a 6 order of difference in order of magnitude of difference between the actual resonance frequency and the differences in frequencies between, for example, the resonance of lipids and the resonance of water. Now, we have to make sure that if we measure, for example, a spectrum in a 1.5 Tesla scanner and the spectrum in 3 Tesla scanner, we always refer to the same units. So we can scale the frequencies by a reference frequency. And the reference frequency chosen, it was chosen to us by chemists that introduced this scaling factor in the 1950s, is that of the least shielded molecule known to them at that time. And this is a molecule called tetramethylsilane, and it was considered to be the one that will give a good reference for all other resonances that are typically less and less shielded depending on where they are in the molecule. And if we scale our resonance frequency by the frequency of the tetramethylsilane, or the TMS, then the resonance frequency of TMS in those unitless numbers called chemical shift in parts per million is 0, and the resonance of lipids will be at 1.3 ppm, and the resonance of water will be at 4.7 ppm, and that is a frequency independent number. And this is again a historical choice, but we kind of stuck to that historical choice. And even today on clinical scanners, we refer to the resonance frequency of water to be at 4.7 ppm and that of lipids at 1.3 ppm. Another historical note is that when we represent those spectra to be measured, for example, on the scanner, or when we prepare our graphs to be sent for publication, then we flip the axes. Instead of going from left to right, they actually go from right to left. We don't have enough time to explain why is it done this way, but this is a kind of a convention where we will see that the numbers increase in ppm and essentially in resonance frequency, going from right to the left. Now that we have everything in order in terms of where our, what is the axis along which we report our resonance frequency and in what direction it goes, then we can go to this very crowded slide and see where are the chemical shifts of protons in biologically relevant molecules. Remembering that, for example, at 0 we have the resonance of the TMS, at 4.7 we have the resonance of water, and then we have a host of other resonances that we can detect in biologically relevant molecules. Now, most of protons resonate in this very narrow range of about 10 parts per million. So from 0 ppm, which is the resonance of the TMS, all the way to about 10 or even to 12 ppm for certain molecules that are actually non-biologically relevant molecules. Most of the biological relevant molecules, most of them, not all of them, resonate in a very narrow range between about 0.5 ppm all the way to the resonance frequency of water. So just to give you an idea, as we saw, for example, the CH3 and CH2 groups of lipids resonate around between 0.8 ppm to 1.3 ppm. But we also have other such CH3 and CH2 groups in smaller molecules that we call metabolites, and we will see them later on. And many of those molecules have, for example, carbons that are linked to nitrogen on one side and to a hydrogen on the other side, and these, for example, resonate between 2 and 3 ppm. We also have many sugars, for example, glucose, or another important molecule that is myoinositol, and these are protons that are linked to carbon, that are linked to oxygen, and they will resonate anywhere between 3 ppm and 4.5 ppm. And then we have the non-saturated fat carbon, so this double link between carbons and the hydrogen that is attached to them actually resonates at a frequency that is slightly higher than that of the water. And broadly speaking, if I may use a pun here, the line width of the resonances will be sort of related to the size of the molecule that you're looking at. So in essence, the molecules that are of interest to us and are easily detectable on the spectrum are those that give rise to narrow lines, whereas the molecules like proton resonances on big proteins give rise to very broad peaks that are very difficult to detect on a standard in vivo magnetic resonance spectrum. And finally, we get to look at how does a spectrum look like when it is taken in vivo from a human organ, and in this case, it will be from the brain. So if we look, for example, at what is the content of any part of tissue that we're looking at, well, most of it will be water. So we will have a very strong water peak that resonates at 4.7 ppm. But in spectroscopy, we have methods to get rid of that signal. And if we get rid of that signal and then focus on the range between 0.5 ppm and 4. Let's say 2 or 3 ppm, then we have a richness of peaks that are related to the resonances of protons that are attached to small molecules that we can detect because they are in decent concentrations in the brain, so between, let's say, 1 and 10 millimolars. And what are those molecules in the case of the brain? Well, some of those molecules are found in neurons. You know that neurons are important cells that are kind of the central processing unit cells in the brain. And two very important such molecules are N-acetylaspartate, which is the molecule that gives rise to the strongest peak in the spectrum taken in the central nervous system or in the brain. It has many different roles that we will not tell about too much now, but one thing is important is that the amount of N-acetylaspartate is modulated in disease. It is also modulated by age, but definitely in disease. So you will see later on diseases in which the amount of N-acetylaspartate decreases because there is something that affects the viability or the number of neurons that we are looking at. Glutamate is the most important excitatory neurotransmitter in the brain, and it gives rise to resonances in about 2.5 ppm. You can see it at 2.3 ppm, and also in 3.8 ppm right here. There are also molecules that are found in glial cells. Glial cells have many different roles in the brain, and one of them that is relevant to our discussion today important molecules are myoinositol, that is a sugar, and it gives rise to resonances around 3.6 ppm, and also choline, that gives rise to resonances at 3.2 ppm. Myoinositol is almost specific. This is important when we are looking at pathologies that involve astrocytes. There are two other molecules that I want to mention. One is the pair creatine and phosphocreatine that gives rise to a strong resonance at 3 ppm, and it is a molecule that is implicated in energy consumption in the brain.
What is
Another
ppm?
molecule that is also implicated in energy consumption in the brain, but also is important from the pathological perspective,
What's
because
good, my
its
nigga?
concentration increases dramatically in pathology, is lactate.
Bloody.
How do you localize our signal? So you know that we use gradients in order
Oh
to
yeah,
localize or
3-slab
to spatially
selection tree.
encode our signal in magnetic resonance and in imaging, obviously. How do we do it in spectroscopy? Well, I'll give you one of the most commonly used tricks in the trade, which is the so-called 3-slab selection trick. Well, we start in a simple and very way that we select a slice in imaging, which means that we give a radio frequency pulse in conjunction with a gradient that allows us to select a slice in imaging.
Excuse me?
And if we apply a 180-degree pulse, we will generate the spin echo. But if we pulse with an operates in a perpendicular
Calm down.
then the echo will
Auntie,
generate
stop it, auntie.
at this point in time
KSI's mom.
only from
Hey, mute
calm down.
yourself.
between the
Mute yourself, please.
Oh, I'm sorry.
Please, everybody mute yourself. Thank you.
You're welcome.
No, I think we're invaded by intruders.
Sorry.
No intruders. We have some nasty figures on the video somehow.
Oh yeah.
Yes, yes.
Yeah.
I'm not even sure what to do with this.
Did they leave or are they still in?
They have intruded. They have intruded our meeting.
I'm going to
Can't do
generate
anything.
another link and share
Who is this?
the group.
Who's James Charles?
Yeah, can you share it? Because these bombers have been like
Yeah, man.
In N-acetyl aspartate, in this big peak, which is the peak associated with neuronal viability and with neurons in general. And there is also an increase in the two glial markers that we talked about before. So there is an increase in choline at 3.2 ppm, and there is an increase also in myoinositol at about 3.6 ppm. So there is a process here that tells us that glial cells are involved. It might be what we call astrocytosis, so increase in the number of astrocytes, but it can also be an increase in activity of those cells that are also associated with the immune system of the brain, indicating that there is an inflammatory process going on. So again, there is an increased choline and a decrease in N-acetyl aspartate. In a, let's say, more famous disease, which is Alzheimer's disease, we see similar processes. So if we look at healthy controls versus patients that have what we call mild cognitive impairment versus patients that have full-fledged Alzheimer's disease, we have a gradual decrease in N-acetyl aspartate and gradual increase in those glial markers, again indicating that there might be an inflammatory process going on, but definitely an increase in number of glial cells, possibly astrocytes, something that also has been substantiated by post-mortem studies. And again, here you see on the graphs, you have a decrease in NAA going from low to intermediate to high cognitive impairment, and an increase in the concentration of myoinositol, or in the signal of myoinositol. And if we take the signal of NAA and scale it by the amount of myoinositol, then we have a kind of a spectroscopic biomarker to the severity of Alzheimer's disease, going from a high number for this biomarker at, let's say, healthy subjects or very low cognitive impairment and going down as the cognitive impairment goes high. So we have a decrease in NAA, which is linked either to neuronal death or atrophy or to neuronal dysfunction, and increase in choline and myoinositol, indicating either glial activation or glial proliferation. So either more glia or active glia, indicating some sort of immune response, possibly neuroinflammation in Alzheimer's disease. Now let us consider a different type of MRS or magnetic resonance spectroscopy experiment, one in which we're not looking at the intensity or the signal strength coming from different metabolites, but to the diffusion property of the same metabolites that we first measured their concentration. So for example, let's take a look at a neuron, and we know that N-acetyl aspartate resides in neuron. And the N-acetyl aspartate will diffuse inside the neuron, essentially allowing us to study the properties of the intracellular space just by following its diffusional properties. And the same thing goes with glial molecules such as choline and inositol. And in order to do that, all we need to do is essentially take our spectroscopic sequence and add diffusion weighting to it. In a very similar way, the diffusion weighted imaging takes standard imaging modalities and equips them with diffusion sensitizing gradients. And in this way, we go from diffusion weighted MRI to diffusion weighted MRS, in which we essentially look only at properties of molecules that are trapped within cells, or either in astrocytes or in neuron, and in a selective manner can say, well, if there is a pathology in one of those type of cells and affects specifically the diffusion properties within these particular cells, then we can add to the explanation that diffusion weighted imaging gives to the picture. For example, in this beautiful study from the group in Paris, where they looked at diffusion properties of metabolites in stroke, well, I give this example just to show you that we have the spectra that you saw earlier before. So for example, in the lower row, you can see spectra coming from a healthy part of the brain, and the strong peak of the NAA at 2 ppm without diffusion gradients is stronger than the peak with the diffusion gradients on. And that means that if we take those two signals, one without diffusion weighting and one with diffusion weighting, we can calculate the diffusivity of N-acetyl aspartate in a very similar way that we can calculate the diffusivity of water from diffusion weighted imaging. Only that we can do it now for all the other molecules for which we have data, for example, to lactate or the creatine or the choline, and each diffusion coefficient of each molecule will give us something related to the milieu in which those molecules diffuse, meaning the cell populations in which they are found. And that means that this gives us leverage to provide information about cell shape and size in health and in disease, but also about, for example, the viscosity of the cytoplasm that again might be modulated by disease. We can also hope to get information from other structures within cells, for example, mitochondria or other structures that may contain specific metabolites. And maybe we can also say something about metabolism, because metabolism may also affect the diffusion properties of molecules that are engaged within a chemical reaction linked to metabolism. And if we have a molecule that has a concentration also outside the cell, for example, lactate is such a molecule, or glucose, then we can maybe have a handle on providing information about the distribution between the intra and extracellular space of those molecules. So in order to say something smart about the link between diffusion properties of metabolites and disease, we have to kind of come up with a hypothesis of how cellular structural changes in disease affect metabolite diffusion. So for example, in Alzheimer's, we know that one of the hallmarks of Alzheimer's disease is the accumulation of a protein called tau in neurons. Or if there is an activation of the immune system in the brain, then cells that are associated with the immune system, be them astrocytes or microglia, change their shape. For example, here you can see non-reactive astrocytes, and when they become reactive, they become bigger, and their processes or fibers become thicker. And the same thing with microglia. It goes from the non-activated shape that is like a star-like type cell to an amoeba type cell when it is activated. And if we try to think about the hypothesis of what will happen to the diffusion properties of metabolites inside those cells in disease, then for example, going from a healthy cell to a tau-filled cell in Alzheimer's disease, one may hypothesize that the diffusion of N-acetyl aspartate or glutamate, they're all predominantly found in neurons, will be disturbed or hindered by the presence of tau, and that the apparent diffusion coefficient or the diffusivity of those molecules will go down. Conversely, we expect the changes in size of glial cells that makes more room for diffusion in the intracellular space to cause an increase in the diffusivity of glial molecules such as myoinositol or choline. And here I want to give you an example for such a study that indeed looks at the diffusion properties of metabolites, in this case in Alzheimer's disease, to link it properly to our hypothesis that we made in the previous slides. So I had the pleasure and the honor to collaborate with a group in Lund University in Sweden, where there is a large study that focuses on Alzheimer's disease. And in this study, they're looking not only using MRI and MRS, but also using positron emission tomography. And there is a possibility to create ligands in positron emission tomography, or PET, that will represent the presence of tau in this case in Alzheimer's disease. And you can see, for example, that on the slide, on the panel on the left, you can see kind of a hotspot of tau accumulation in an Alzheimer's patient in the area of the brain called posterior cingulate cortex. And this is what we are really interested in. So we are looking at the area that we know that there is tau accumulation in Alzheimer's. And what we will do is we will study whether there is a change in the diffusion properties of metabolites in this particular region, and if we are able to link it to the accumulation of tau. And to our, let's say, pleasant surprise, we indeed found that if we're looking at the apparent diffusion coefficient of N-acetyl aspartate in Alzheimer's disease, so going from a group of healthy subjects into a group of patients with Alzheimer's disease, where we know that there is an increase of intraneuronal tau protein, we indeed measured a significant drop in the apparent diffusion coefficient of N-acetyl aspartate. And this is in strong contrast to the increase in the apparent diffusion coefficient of water exactly from the same region. So we can say that the apparent diffusion coefficient of NAA is lower in the posterior cingulate cortex in AD, possibly because it is linked to tau accumulation. Whereas the ADC of water in that very region is higher than in healthy subjects, possibly because of increase in amount of extracellular space or in increase in the rate of water exchange between cells and the extracellular space. But you can see that these are two measurements that are independent and report two opposing trends, which means that we're looking at two processes. And this is, I think, a beautiful way to exemplify how the diffusion of metabolites complements in information the information that we get from diffusion of water in the same patients. And now to the last topic that I want to talk about today with you, which is the possibility of having low field MRS. Now, I'm sure you saw this slide many times, not only today, but in other presentations, and I will not even dwell on it because I think we all agree that the contribution of low field worldwide is potentially enormous. And the only question that I want to ask is there a role for low field MRS? And I hope to convince you that maybe in some time we will be able to introduce a role for MRS in low field. And let's try to do it together now with the next couple of slides. So first of all, we need to select a target for spectroscopy at low field. There are many reasons why I chose as a target lactate, but one of the main reasons is that lactate is a molecule that in the healthy brain is at very low concentration, but in the diseased brain, the concentration of lactate increases quite dramatically. And here are two examples. One is in stroke, where you can see the presence of lactate at 1.3 ppm, where you don't see it in healthy parts. You can see it here in tumor, a very strong presence of lactate, which has a very interesting fingerprint that we will mention a little bit later on, of two peaks that are separated by a very well-defined frequency, and it is a frequency of 7 Hertz that I will mention later on as of its origin. To me, one of the most interesting and most, I should say, critical ways of using spectroscopy in general and in potentially also in high field is to look at what is called hypoxic ischemic encephalopathy in babies. And these are babies that are essentially born without enough oxygen during the birth process. And that gives rise to an increase in lactate because of anaerobic metabolism. And this amount of lactate is high enough to detect. And not only that, but lactate becomes a very important marker, not only for the degree of pathology of those babies, but also at the prospects of their healthy development later on, as mentioned, for example, in this particular quote that you can see here down below. And now to the challenges of spectroscopy, and in this particular case of spectroscopy of lactate at low field. So let's start with spectroscopy at high field. At high field, the lactate signal, which derives from this molecule, gives rise to two distinct resonances, one at 1.3 ppm and one at 4.1 ppm. And those resonances, as you can see, are split into several lines, 2 of them at 1.3 ppm and 4 of them at 4.1 ppm. The separation or the split of those peaks is due to an interaction between the protons that generate the 1.3 ppm, those are the 3 protons circled in green, and the proton that resonates at 4.1 ppm, the one circled in purple. This is an interaction that we call spin-spin interaction, or J coupling, or scalar coupling, and it gives rise to these split lines that give rise to this very distinct spectral contribution of lactate that you also saw in the previous slides in vivo. But what happens at low field is that the chemical shift between the 4.1 ppm and 1.3 ppm becomes so small that all of those peaks coalesce into one broad contribution. That means that all the spectral information within the lactate, but also between the lactate and everything else, including water, including N-acetyl aspartate and all the other metabolites that contribute to the magnetic resonance spectrum, all this spectral information is masked, giving rise to inability, essentially, to perform standard spectroscopy at very low field, especially with the type of magnets that we are using in our low field MRI systems. But to the rescue comes a very old physics, MR physics principle. And this is the principle of taking spectra out of a series of spin echoes, one after the other, using the so-called Karp-Purcell-Meibom-Gill sequence, which is a sequence of 180-degree pulses following the excitation that gives rise to consecutive echoes that come one after the other to generate what we call an echo train. The signal along this echo train decays according to the relaxation constant that we call T2. And if we take each summit of each echo and concatenate them into a time series, and we do a Fourier transform of this time series, well, we will get a signal that has no oscillations and will give us essentially information about the T2 decay of the resonance at this frequency. However, in a J-coupled system, like for example lactate, the echo train is modulated, meaning it gives an additional oscillation in addition to the exponential decay. And when we do a Fourier transform of this signal, we get a spectrum that doesn't have any of the chemical shift information, but has the information that is stored in this interaction between spins that we called the J coupling. And this gives rise to what we call a J spectrum. So when I was in Leiden a couple of years back, Andrew and I had this idea to try to do an experiment in which we collect J spectra from phantoms, or essentially, as you will see, ping pong balls that contain different solutions, including a solution of lactate. So here is the prototype of the low field MRI spectrometer or imaging apparatus that Andrew built together with his team in Leiden. And together with the team, we generated a radio frequency coil. Tom O'Reilly built this beautiful coil, and we had ping pong balls. There was an idea of Andrew to fill them up with solutions of lactate. We will also show some results in the phantom that I show here below, but essentially the sequence that we use is a very simple sequence. It's a Karp-Purcell-Meibom-Gill in which we collect those echoes and we try to concatenate them and then see what kind of oscillations are present in those echoes representing the lactate molecule. But before we did those experiments, we simply collected a spectrum from the lactate ping pong ball that is indicated here by the red arrow and tried to see whether, whether we can see any spectroscopic fingerprint that separates the lactate, for example, from other molecules present, in this case, water. And we could not see anything. All we got is this kind of blob of a peak that doesn't have any spectral characteristics whatsoever that can distinguish between anything that is present in this phantom. But when we performed our Karp-Purcell-Meibom-Gill experiment, that, just to remind you, is expected to give us a modulation on the spin echoes that will indicate the presence of lactate in the sample, we did obtain signal that you can see very clearly does not simply decay in an exponential way, but has an additional modulation to it. And this is exactly the modulation that we were looking for, that would be an indication that we have a J-coupled system within our phantom, and following the Fourier transform will give us those spectral patterns. Now, the theory behind how the oscillations in the decaying echoes is related to the signal that we see from lactate is quite complicated. But I hope that I can convince you that those spectral patterns are quite unique, and unique enough that if we can detect them in the concentrations that we expect to see in lactate in vivo, in the diseases that I mentioned earlier, those spectral patterns can be indeed a biomarker at low field for the presence of lactate in disease. We even went one step forward and created a phantom that has 4 containers with 4 different concentrations of lactate, and we're able to image that signal, so to take our spectroscopic measurement and to so-called spatially encode it. So we now have an image of the concentration of lactate in 4 containers going from 0.75 molars to 1 molar to 0.25 molars and 0.5 molars of lactate. These are very high concentrations, so this is a kind of a proof of concept alone, but we intend on trying to develop it in such a way that we will also be able to apply to lower concentrations. So to conclude my talk, magnetic resonance spectroscopy is complementary to MRI in the sense that MRI gives us a very highly resolved but non-very specific view of tissue, in this particular case of the brain, whereas magnetic resonance spectroscopy, which is less sensitive, provides information about aspects that MRI cannot provide us information about. For example, biological processes and different pathological processes that affect specific types of cell populations. We also saw that we are not just interested in concentrations in spectroscopy, and other metrics or other derived quantities can provide very unique markers to pathology, in which case if we combine this type of measurements with imaging measurements, we have a broader and more explanatory picture of the pathological processes that we see in different diseases. And the last thing is that with some tricks of the trade, MRS at low field is possible. There are still many challenges that remain to make this to work in vivo, but this is why all of us as a community work together to develop these methods that hopefully both in imaging and in spectroscopy will provide valuable information at low field on different diseases that I hope you will be able to apply and to investigate together with us. And with this, I'd like to share with you some pictures of the city in which I am, which is Brighton, and to thank you all for your kind attention. And I'm very, very glad to take questions either in real time or offline. So please ask me whatever you guys are interested in. And again, thanks very much for inviting me and to having me on this wonderful workshop.